Okay, thank you. Okay, our next speaker is Professor Paul Herrling. He is the chairman of the board of the Novartis Institute for Tropical Diseases in Singapore. Before that, he was head of corporate research at Novartis. He now is retired as myself, but he still serves on several boards of universities in Switzerland and on boards of many other organizations. He will give us an update on the situation of malaria drug discovery. Please, we are looking forward.
Thank you, Professor Kuhn. Good afternoon. So, some of you were here last year, and I started to give you an overview of the work that we're doing in Singapore to try to address the point that Professor Brun was saying, the need for new tools in order to reach the goals of elimination in some of these diseases. So I had presented the first project. I will quickly summarize it afterwards, and today I will give you an overview of a second tool that we are developing. As you know, in all of these kinds of bacterial diseases, but applies also to parasitics and viruses, one drug is never enough. Certainly not in the early pipeline, because you have a very high attrition rate and you lose most of the drugs in the development, but also for the point that was already made by Professor Brun, namely resistance. And you need in most cases today, you would always combine drugs to attack a parasite or a bacterium. It's actually very dangerous to use monotherapies. So I'll talk about malaria. The next slide will just give you a little bit of the dynamics of malaria from 1900 to now. So you can see here 1900, 1990, 2016, and again the goal here is the elimination by 2040. So the malaria community has given themselves 10 years more than what we saw just before. So that's actually quite impressive. You see that malaria was nearly everywhere in 1970, and how it did shrink through many measures. Many of them were things like getting rid of still water and stuff like that. Okay, now there has been quite some progress since 2000. You can see we estimate between 2000 and 2015, through all of these measures, prevention, bed nets, and drugs, you could prevent about 6 million deaths. You can see that there was a reduction in the incidence by nearly 40%, and in global mortality are about 60%. And still, we still have about 214 million cases of malaria, mostly in sub-Saharan Africa today, or in 15, and still about 400,000 deaths. So drugs, medicines account for about 22% of the reduction that I've shown here. So obviously there are other measures too. The reason why we constantly need new drugs, and we had the Artemisinins, which are currently the standard therapy, an artemisinin fixed combination with lumefantrine or with other drugs, which is really a very potent tool against malaria. It was introduced around 2000, but now 16 years later we have this problem. And that I mentioned it last time also is that you can now see the emergence of parasites that are resistant to artemisins. And that, by the way, relates to what I said before, and that has probably to do because there are, and I stressed that last time also, what I call criminal companies who are selling the artemisins as monotherapy. And this occurs a lot here, but also in Africa. And you can see that the red part is the proportion of artemisin-resistant people that you can see. Of course, it's the parasites that are resistant. And that was what started our program 9 years ago at the urgence of the Wellcome Trust to try to make new tools. And our institute in Singapore is geared, actually it is also a public-private partnership, Professor Brun, because it is, I'll show you, it's a collaboration with the Singapore government. You forgot it, but anyway. So, okay, so that is a quick reminder of the cycle, very simplified, injection of the parasite into the patient's blood. It goes into the liver. Up to here you have no symptoms. Then it goes into the blood. This is when you have the symptoms, and it can be very dangerous. The cerebral form can kill you. And then you go into the sexual form, and these gametes, the sexual forms, are the ones that are being sucked up again by the mosquito and redistributed. So what we need in those new tools is certainly we need novel mechanisms, because we want to overcome all the known resistances. So any of the mechanisms that have been used up to now must be avoided. So novel mechanism is key. A second very important goal is to try to make faster acting drugs, because we see today the artemisinin combinations take about 3 days to eliminate the parasites, and that already produces compliance problems. So if we could have a pill that does it in once, this would be extremely useful. Okay? Then we also would like to have drugs that can kill the gametes here, because if they do that, we cut the transmission cycle. Then, because I said the symptoms, once the symptoms are there, they can be dangerous, you would like to have drugs that are prophylactic, that kill the parasite before the symptoms emerge. Then the thing that maybe I'll talk about next year is the ViveAct. So I will talk now mostly about a drug that does this and this, but not the single dose. Now, that's what I was saying. You can only do this with very intensive partnerships. That's here. The leader of this project is the Novartis Institute in Singapore. But we have a large number of partners, both within the Novartis research family, which are really very important, but also at other companies, at universities, both in the US, in Europe, and here you can see in Asia. And of course, the Swiss Tropical Public Health and Tropical Institute is one of our key partners also. This is funded by Novartis, by the Wellcome Trust, the Medicines for Malaria venture you saw before, and the Singapore government. So the key of the success, what I'm talking is what last time and this time is only 9 years, which in a drug discovery cycle to reach phase 3 is extremely fast, especially in an area where you have to start from scratch. And one of the reasons that we could be that fast is that we were able to create an assay. This is absolutely key, to have an assay that can be run automatically. So where you can run millions of compounds against the assay. But of course, the assay needs to measure something that translates into the human situation. Of course, you will always be able to measure something, but you need to measure something that is relevant for the patients. In this case, it's the parasites, which a green fluorescence protein attached to an inside, and we see them inside human red blood cells, which is where they live when they create the disease. And so with the green fluorescent protein, we can see are there parasites or not in these wells. So it's large numbers of wells, automatic imaging, and we can see where they die and where not. And that's how we select the starting point. So that was extremely important to very quickly arrive at hit compounds, and then chemistry comes in, makes leads, and makes the actual drugs. I won't go through that process again today. Now, last time I talked to you about this compound, which is the fastest parasite clearer we've ever seen in about 10 to 12 hours, and has the possibility maybe to be such a one-dose cure or one-day cure that I mentioned before. It is now in phase 2b. We're just looking now at most of the malaria compounds have some effect on the liver, this one too, and we need to explore, is it dangerous, is it not dangerous, what are the doses and things like that, and the combination, we need to find a combination partner with this, because as I said, we're not going to use it alone. What I'm going to talk to you today in focus is on the second compound here, which is in the clearance time is again in the same range than the artemisinin, the current compound, so lumefantrine. But the difference between this and this This one affects only the blood stages and the gametocytes, whereas this one hits also the liver stages. That is, you can kill the parasites in the human body before the symptoms occur. So this is why we say, that was the 609. We also discussed last time the target, which is specific ATPase, the Plasmodium falciparum ATP4, and which is a sodium pump. Here, what we have is, as I mentioned, something we could use for prophylaxis because it kills the liver stages. But it's the story of the target, you will see, is a little bit more complicated than in this case. So first, for those of you pharmacologists and chemists, that's the actual structure. It's different than 609. This was 609 was a spiroindolone. This is an imidazole piperazine. And here you can see one of the animal models we have, which are extremely useful and fast because they're non-invasive. Here you can see the injection, a mouse has just been injected with a parasite. You can see it's, again, green fluorescence protein marked parasites, but we have an imaging system that actually can see the green fluorescent protein throughout the skin inside the body. And so you can see here after infection, after a few hours, 24 hours, you can see the liver infection. And then from the liver, it grows out, and then it's most of the body, including the brain. If you use this compound, this new compound, and you apply it even before, you can see you can completely prevent the infection and the spread through the body. So it's a new chemical entity. We know it is none of the mechanisms that we know before, so the criterion of novel mechanism is fulfilled, but we don't yet know exactly what it is. I will show you why. We're a little bit unsure about the mechanism. This is the proof of concept now in man. These are patients. And what you see here, this is actually ViveAct. The ViveAct parasite gets killed also pretty fast in 24 hours. What you see here is hours after the infection. And here you can see parallel parasite levels. So you see this line says when most of the parasites, 90% of the parasites have been eliminated. So it's 24 hours in ViveAct. It's 45 hours at 400 milligram dose here. And interestingly, when you increase the dose, which did also to looking at safety and things like that, you don't get any faster action. It's about the same. So this is the evidence in people to show, one, it does eliminate the parasites, two, it takes about 2 days. Now, when we did the same mutation studies that I described last time with 609, where you treat the parasites with sublethal doses of your new drug and then select mutations, resistance, and then see where in the genome you can find mutations. In the 609 case, in the previous, you remember, you could see all the SNPs in only one single gene, which was this ATPase that I mentioned. Here you find some in this thing, it's called PF-CARL, which is Plasmodium falciparum cyclic amine resistance locus, which is usually these names is when you don't really know what it does. But all the SNPs were in one study were here. There have been in the literature, however, already for these imidazole other types of mutations that have been described. And here I will show you another one in a second. Important is also that this resistance is relatively moderate. That is one resistance per 10 to the 8th parasites, which is good. So there's not an easy resistance. And the shift in IC for this one, it is 20 to 700 fold. So that is, parasites that have these mutations are about 700 fold more resistant to the drug. Now, this is why I say here it's not so clear. Looking further, one could start finding also mutations in these resistance experiments in other genes. So the UDP galactose transporter or acetyl coenzyme A transporter. And at the point where we are today, we don't yet know in nature which are the ones that would come first. And so we now also have to continue studies to select is this compound affecting obviously all of these targets, and how do they contribute to the biology and to the medicine of the compound. See, this is 300-fold shift, this is 400-fold shift. So the strongest was the Carl that I mentioned about before. Okay, so you see that with these methods that I described last time and today, we can now, with the modern methods of high throughput and robotics and non-invasive techniques, we can now, for malaria, that does not apply for other parasites and bacteria or viruses, we've been able to find quite a number of different new genes that are targets for drugs. And this is what I mentioned before. And by the way, here again, this compound 156, like 609, affects also the gametocytes. So both of the new compounds that I've been describing last time and today will be transmission breaking. So these data have that in common. This new compound has the advantage of, as I mentioned, to hit the liver stages. I described these new non-invasive animal models that you can use and which are quite predictable. And also, of course, the necessary models that you need to have before you go in man, safety, PGA, and PD. So to summarize, you can see that the compound I discussed last time had that was this target, which is new. I talked about this PF Carl today with 156, which is also new. This is, by the way, to show you attrition. This is a dead compound that we had hoped to develop for ViveAct, but it turned out to be too toxic. But also showed us a new target that was not known before, and now we're trying to use this target to find more compounds. This is the Artemisinins. And many of those here, these targets are new, have never been described before, and they're all different from the classical antimalarials. So they should overcome, they actually should overcome the resistance. So I want to leave enough time for discussion, as Professor Brun asked me before. Here, so in summary, you can see how leveraging mechanistic data and all of these modern technologies, we are now building a whole array of tools against malaria, because we will then have several that we can combine in order to get to this goal of eradication or elimination that we talked about before. So, and for the falciparum, as you've seen, we are much further ahead. I'll discuss this one probably next year. It's very difficult. That is Plasmodium vivax, and it has the numbers. The figure I showed you before, where you could see vivax parasites eliminated in patients. This was the blood form. The problem in vivax is that this form here, which is called the hypnozoites, lives in the liver and sleeps, as the name says. And when all of the blood parasites have disappeared, this eventually wakes up again and creates a new cycle of malaria in a patient that has not been reinfected. It's just a dormant form that wakes up. And the problem is we have no good high throughput assay for this form. It's extremely difficult to study this thing. I'll talk about this maybe next year. So this again, just to remind us that how many people contributed to this kind of work. With that, I'll thank you for your attention, and I hope we'll have some time for discussion later. Thank you.
Thank you very much for this inspiring presentation. I'm sure that there will be lots of questions, and I'm looking forward to the discussion. Now, I see the boss coming.
Thank you very much for this presentation. How will you avoid next time the problem of monotherapy and resistance developing? So my question was how to overcome next time the development of criminal resistance induction.
The best way is combination.
Okay.
But, of course, otherwise you would need armies to get rid of the people who sell the monotherapies.
Okay.
That's a political problem.
Thank you. We have the questions at the very end all together, you know.